Hello, that's my great pleasure to open this session on the second session of this workshop on effects and risk. So I'm Dominique Laurier. I'm the chair of the committee one, which is dedicated to the health effects of exposure, health effects and biological effects of exposure to ionizing radiation. So we are going to have a 2 hour session with 5 speakers, John Matthews, Nobuo Koban, Jacqueline Garnier-Laplace, Yuichi Tsunoyama, and Klaus Strotz. So each of the presentation will be about 12 minutes, and we are going to have one or two burning questions just after that. This organization should leave approximately half an hour of discussion, questions and answers at the end of the session. So I'm going now to introduce the first speaker, so John Matthews. John Matthews is an experienced epidemiologist and is recently affiliated with the University of Melbourne in Australia. He led the large record linkage study defining cancer risk in young Australians exposed to low-dose radiation from CT scans. And today we'll comment on plans to use national record linkage to study next generation effects of ionizing radiation. So John, the floor is yours. The floor is yours.
Why should we think about next generation effects? Well, it's almost 100 years since Muller showed that ionizing radiation could cause germline mutations in fruit flies. But we still do not know whether germline mutations attributable to low-dose radiation have an adverse effect in human populations. We've been thinking about this ever since the time of the atomic bombings in Japan. And of course, no extra mutations were observed in the children of survivors. Many other concerns have been heard over the years. For instance, the cluster of leukemias amongst children of radiation workers in C-scale. And of course, we all know the Gardner hypothesis was discredited. And in many populations today, medical procedures now deliver as much radiation as comes from all other sources combined. So if we do think about medical imaging of parents, we wonder whether it can contribute to adverse outcomes in the next generation. And if that's possible, we need to urgently reassess risks and benefits. What do we really know about the effects of ionizing radiation? What do we know about the mutations? Well, many of you will know that recent molecular work has identified single nucleotide variants, point mutations, which are frequent, around 44 to 82 new SNVs per generation, mostly of paternal origin. They increase with paternal age, and they're mostly neutral and rarely cause disease. These are contrasted with copy number variants, deletions and duplications, which are much less frequent, 1 to 2% per generation, but very large in size, ranging up to 10 megabases. And they're much more likely to cause disease, especially if large. They're subject to purifying selection, and they can cause intellectual disability, congenital abnormalities, and cancer. And there is quite a bit of evidence now to show that CNVs can be caused by ionizing radiation. So when we think about next generation effects, are they fanciful or not? I'm suggesting they're not fanciful. We know that genetic mechanisms to repair radiation damage to DNA are highly developed, and we take that as evidence that radiation is an important agent of natural selection. We know that germline mutations, especially deletions and duplications, copy number variants, contributed to outcomes such as intellectual disability, congenital abnormalities, and cancer. We know that low-dose radiation contributes to both germline and somatic mutations, and that CNV are much more important than SNV in causing adverse effects. And we really are at the situation where we know that radiation-induced mutation in humans needs to be better understood. We also know that radiation in pregnancy is known to increase the risk of cancer in the child, but this risk has not been adequately re-evaluated since the introduction of CT scans in the 1980s. So we need to quickly think about the new germline mutation study after Chernobyl, first author, Jaeger, published in Science earlier this year. And what they did was whole genome sequencing of 130 children and their parents, that is trios, to identify de novo mutations following parental exposure to Chernobyl radiation. Well, to cut a long story short, they recovered de novo mutations. They were almost entirely single nucleotide variants, and a total of a mean of 90 mutations per child, of which 72 were single nucleotide variants, 18 others, and they weren't able to measure copy number variants, and they found no increase in mutations with parental radiation dose. So that's basically saying that point mutations were not increased by parental radiation after Chernobyl. And of course, they weren't measuring phenotypes, they were just measuring the genes. Now, why might previous studies have failed to detect next generation effects of radiation? Well, we know that deleterious effects will be rare, so that a large number of exposed individuals are needed to achieve power. Single locus studies will be of very low power. It would be better to accept group phenotypic effects at a given dose with the whole genome as the target. Deleterious syndromes that are of early onset may not survive to be enumerated even in large studies, and some might even not be recognized as genetic in the era before genomic sequencing. So it's possible that germline CNV mutations at low radiation doses are not seen at higher doses because of selection against damaged gametes. And of course, that would imply that dose responses are non-linear. And of course, it's possible that any real effects of parental radiation are so small as to be negligible. And of course, that was what most of us have been thinking in recent years. Can the next generation question be answered? Well, the opportunity is also the challenge, because there are large numbers of people who are exposed to diagnostic medical radiation. We can pick that challenge up by using medical records to estimate radiation doses for exposure before conception or in pregnancy, and we can use record linkage to assess next generation outcomes in offspring. We can test whether outcomes increase with parental radiation doses prior to conception or in pregnancy. And if we think about mutation rates, target sizes, and power, if we're only looking at a single locus, we would have very low power to detect any effect. But the essential question we're interested in is what's the overall deleterious effect of a given dose of radiation if we consider the whole genome to be the target. By grouping similar phenotypes together, we have more outcomes, giving us greater power to reject the null hypothesis of no effect. For example, if we had 5 million children, of whom 2% have intellectual disability, say, for the parents who've got linked radiation histories, there'd be about a history of radiation in the year before conception in about 10%. So that if the null hypothesis were true, we would expect basically 10,000 affected children to have a history of parental exposure. So that, for example, gives a great power to detect a 5% increase in adverse outcomes attributable to parental radiation. So in summary, there is continuing uncertainty about the importance of ionizing radiation as a cause of harmful germline mutations in humans. It's important to measure the magnitude of any next generation risks because medical radiation contributes substantially to individual radiation doses. We already know that ionizing radiation can cause de novo copy number variants, which are predictive of adverse outcomes, such as intellectual disability, congenital abnormalities, premature mortality, and cancer. We've got a plausible causal pathway that starts with low-dose radiation and ends with de novo CNV mediating adverse effects in an affected offspring. If this pathway is important, and that's a big if, there should be a correlation between the timing and radiation doses to parents and the risk of genetically mediated adverse effects in offspring. To test this potentially important pathway, we've planned a national record linkage study to estimate parental doses of medical radiation and to assess whether doses before conception or during pregnancy are predictive of adverse outcomes amongst some 7 million Australian offspring. Thank you.
Thank you very much, John, for this very interesting and comprehensive presentation. I can't see any open question for you. Maybe I can use Q1. So your point is about the trio studies, the trio study that has been performed on the liquidators from the Chernobyl accident. In my view, this kind of study is a very interesting compromise between epidemiology, bringing together epidemiology and biological information. You seems not to be so kind with the conclusion of the authors. Can you give more details about that, or extend a little bit about that?
Well, it's an important study, but I'm actually suggesting that looking at the point mutations, which is effectively what they were doing, was looking at the wrong measures because most point mutations are selectively neutral. They don't cause disease, whereas the copy number variants are the ones that cause disease. And if we're looking at mutations because we're interested in adverse outcomes, then we should be looking at the copy number variants rather than the point mutations, the single nucleotide variants.
So I know maybe we can take a second one before going to the next presentation. There are several questions going on now. The first one is, what time period will children be followed after the study? So I guess it's about the study you propose.
Well, what we would do, we'd ascertain 7 million children from the time they were born, and we'd be able to look at outcomes happening up until the present time. So the people, the children from earlier on when radiation records, health records were available from 1985. So we'd be looking at all births since 1985 and looking at the parental radiation during pregnancy or before conception. And the outcomes would be from 1985 to the present. So we'd only have short outcomes on the recently born children, but we think that the adverse effects that are most important will be the ones that appear in early life anyway. Yeah.
So there are additional questions, but I think we will come back to this question of potential effects for offspring at the end of the session. And indeed, the ICRP considers it's a very important question, and C1 is a task group in Committee 1 is under preparation on both effects following in utero exposure and preconceptional exposure. We will come back to this point clearly at the end of the session. I would now move to the next speaker, which is Nobuhiko Ban. So Nobuhiko Ban is a commissioner at the Nuclear Regulation Authority in Japan. He joined ICRP Committee 1 in 2013 and switched to Committee 4 in 2017. He has been chairing the task group 102, which was dedicated to radiation detriment calculation methodology. The aim was to reproduce the calculations that have been done for publication 103. And so, Nobuhiko, the floor is yours.
Yeah, thank you, Chair, for your introduction. So let me share my display. Okay, my talk is based on the work of ICRP Task Group 102. Co-authors are the core members of the group who drafted the report. Task Group 102 on radiation detriment calculation methodology was established in 2016. Its mandate is to detail the detriment calculation in publication 103 and to identify potential improvements in calculation procedures. The outcome is expected to form a solid basis for future recommendations. The procedure for calculating radiation detriment has two major parts. The first part is nominal risk calculation, which is an estimation of the lifetime risk from radiation exposure. Risk estimates of cancer are averaged across sexes, ages at exposure, and geographical populations. The second part is severity adjustment that takes into account lethality, quality of life, and years of life lost. The current scheme of detriment calculation is carefully designed, but it needs to evolve according to advances in healthcare and scientific knowledge. The task group identified areas for improvement as listed here. In the following slides, I will outline what needs to be done for each of them. Calculation of radiation detriment requires baseline cancer incidence rates and mortality rates of reference populations. Baseline rates used in publication 103 are those of the 1990s and need to be updated. It should also be noted that incidence and mortality vary considerably around the world. Inclusion of populations other than selected Asian and Euro-American would provide a broader representation of the world population. For severity adjustment, 3 parameters are used: lethality fraction, quality of life factor, and relative years of cancer-free life lost. Lethality fractions are derived from US population data dating back to 1950s. They need to be updated based on recent data, including those outside US. The same applies to relative years of cancer-free life lost. The quality of life factors are determined as judgment-based values, and they should be revised in a more objective manner. This may further lead to a review of the way of adjusting severity. More universal metrics such as quality-adjusted life year, QALY, and disability-adjusted life year, DALY, are now available to estimate and characterize the QOL for a wide range of conditions. The use of these metrics may be helpful to make the severity adjustment simpler and more reliable. Risk models need revision as well. Radiation-associated cancer risk models were mostly derived from the lifespan study of atomic bomb survivors. Updated models with a longer follow-up are available. Current risk models take into account effect modification by sex, age at exposure, and attained age. Consideration of other modifying factors, such as cigarette smoking, will provide a more accurate picture. Specific risk models are employed for 10 cancer sites, and the remainder category accounts for about 20% of the total radiation detriment. More cancer site-specific models should be developed to reduce the contribution of this heterogeneous category. It may be worth looking to risk models that are not from the LSS. During the last decade, models based on other epidemiological studies were proposed, especially for protracted exposures. The risk models rely on several assumptions, including the LNT model, application of a DDREF, and the risk transfer scheme based on the weighting of ERR and EAR models. The validity of these assumptions must be examined in the light of the latest scientific findings. Finally, revision is also necessary for the risk of heritable effects. It is desirable to review the up-to-date literature on the mechanism of inheritance, available data, and the methods for risk estimation. Radiation detriment is averaged across sexes. This figure shows radiation detriments calculated separately for each sex, as well as the sex-averaged values. Even excluding female-specific cancer sites, ovary and breast, a marked sex difference is found for colon, liver, lung, and thyroid. Radiation detriment is also averaged across ages at exposure. This figure compares the difference between three groups of age at exposure: 0 to 14 years, 18 to 64 years, and 0 to 89 years. You can see the detriment is highest for the young population, except lung. For stomach, breast, thyroid, and other solid category, the difference of more than 2 times is observed. To better handle the variation illustrated in the previous slides, we propose to calculate lifetime risks separately for sexes, age groups, and possibly for other influential factors, and then average the estimates of each in the last stage. The former part is a science-based risk assessment, while the latter is the integration of information for radiological protection purposes. This approach makes clear distinction between them and will provide a better understanding of the construction of radiation detriment. In this context, additional consideration might be necessary for exposure scenario. For example, children, pregnant women or elderly may raise special attention in post-accident situations. Scenarios of protracted exposure over a certain period of time may be worth considering in this regard. Calculation of radiation detriment involves many steps and various information. As the methodology becomes elaborate, a full description of the calculation steps is necessary to ensure transparency and traceability. Ideally speaking, an open source software to perform the calculation should be developed. Radiation detriment basically takes the form of an expected number of cases among an exposed population. However, the severity-based weighting makes it difficult to interpret the resulting numbers and also incompatible with the risk assessment for other hazards. So it is desirable to reconsider the way of expressing detriment. This may include the use of an alternative to the radiation detriment, such as DALI. In any case, it is not straightforward to express the multidimensional nature of the detriment. Efforts should be made to better communicate it. For example, illustration of the data on reference populations will be useful background information. In recent years, there is accumulating evidence that diseases of the circulatory system, DCS, and cataracts might be induced at much lower doses than previously thought. ICRP recommended a threshold of 0.5 gray for these effects in publication 118, but there is considerable uncertainty about the shape of the dose-response curve at doses below this value. It has been discussed whether these effects are tissue reaction or stochastic effect, but the indecisive situation prompts us to revisit the validity of this classification. Even if the dose response is determined in some way, the challenges don't stop there. Statistics on baseline incidence rates are limited and not as standardized as those for cancer. Adjustment for severity could be complicated, considering the large variation of symptoms and conditions. Finally, we are not sure exactly what the target tissues for DCS are. Under such circumstances, whether or not to include these effects in the calculation of radiation detriment remains an open question. Committee 1 of ICRP has already initiated efforts to address the identified issues. The dose response at low doses and the DDREF have been reviewed by Task Group 91. Consideration of DCS from radiological protection perspective is a key challenge, and the dedicated task group TG119 has been established. Reviews of other issues are also getting started in subgroups of Committee 1. Some of these task groups and subgroups provide on-demand presentations in this digital workshop, and you will find out more details. Thank you for your attention.
Thank you very much, Nobuhiko, for this very good presentation. I think we have time to take one question. There have been one or two already on your talk. The first one was from Joachim Breco. The question is, I think the concept of detriment is a good example for what the philosophy of radiation protection should be. Do we need more refinement to make radiation protection more accurate, or do we need less refinement to make it more comprehensible? What is your opinion, Nobuhiko, about that comment?
Actually, detriment, and specifically radiation detriment, is a quantitative basis of radiation protection. So it should not be changed drastically. We need to improve it and to make evolution in a desirable way. So I presented the possible ways of improvement in my presentation.
And maybe the second question, and we will move to the next speaker. Question from Thomas Jobert. Do you agree if we describe the radiation detriment as a risk management tool? It's difficult to say it provides a risk assessment. This point seems very important when we communicate about this concept.
Yes, it is very important point. Actually, originally, radiation detriment concept was developed for ICRP itself, but it could be used for in some decision-making process, for example, for the practice of optimization. But we need to emphasize this is not a tool for scientific risk assessment. For assessing risk for specific individuals, we need to use more customized risk models and realistic assumptions.
Thank you very much, Nobuhiko. I think it's time to move to the next speaker. So the next speaker is Jacqueline Garnier-Laplace. Jacqueline is Deputy Head of the Division of Radiological Protection and Human Aspects of Nuclear Safety at the OECD Nuclear Energy Agency, the NEA. She joined the agency in May 2019 and serves as the Scientific Secretary for the Committee on Radiological Protection and Public Health, the CRPPH. And previously, she was a Deputy Director in charge of research on radiological protection at the French Institute of Radiological Protection and Nuclear Safety, IRSN. And she's currently the Scientific Secretary of the ICRP Committee 4. So Jacqueline, the floor is yours.
Here I am. So hello to everyone on the web. Let me first start by thanking the ICRP and the Program Committee of the Digital Workshop for the invitation to present our work live. Today I will discuss why and how to reduce the uncertainties related to radiation-induced health risk that remain in the area of low dose and low dose rate exposures. I will do so by putting this issue into the perspective of the work of the high-level group on low-dose research that was recently established by the Nuclear Energy Agency. Let me start by briefly introducing the NEA. The agency is an intergovernmental body that facilitates cooperation between its now 34 countries to pursue excellence in many areas, such as nuclear and radiation safety, technology, policy, regulation, and practical implementation. Actually, the strength of the NEA lies in its 8 topical standing technical committee, one of them being the so-called CRPPH, the Committee on Radiological Protection and Public Health. And around this table, we've got together senior top officials and technical specialists from NEA member countries and beyond, with strategic partners to solve difficult and emerging issues and to establish, for example, best practice and obviously promote international collaboration. So let's jump into the core of the topic. Why is there a need to reduce the uncertainties about health risks in the low dose rate area? To make the story short, I like to use this graph from Unscared to summarize the situation in a simple way. This graph represents the plausible dose-response relationships for the excess risk of cancer in the ranges of very low, low, and moderate doses. The data points and confidence intervals on your right-hand side represent observations of increased frequency of occurrence of a specific cancer type in populations exposed to moderate doses. These data come from, for example, from the Japanese lifespan study. So the different lines represent different dose response model for inferred risks of cancer for exposures in the range of low and very low doses, which is actually the most common situation in exposed population, either the public or workers. So recent reviews of biological and epidemiological data tend to provide additional support to the assumptions that for low dose rate exposure, stochastic effects follow a dose response with no threshold. But, and there is a big but the linear non-threshold or the LNT model used for radiological protection purpose, mostly derived from the model fitted on the LSS data, remains still controversial due to uncertainties in this area. Those uncertainties mainly arise from the need of three major assumptions to apply the LNT model in a different domain than the one where the model was fitted, defined by the LSS data. First, transposition from the LSS data to other populations. Second, extrapolation from moderate high doses to low dose. And third, assuming the analogy of mostly acute external exposure to chronic exposure. So in a nutshell, although lots of research have been done and still continue to be funded across the globe, open questions remain. Controversies on the assumptions necessary to assess radiation-induced risk at low dose or low dose rate. We are sometimes facing also discrepancy between results from radiobiology and epidemiology. We know that risks vary according to individual characteristics and that more research are needed for a better understanding of this. And in addition to cancer risk, uncertainties also apply for non-cancer risks. And as it has been reminded that by the first speakers, also for heritable effects. So this situation impacts policy, impacts regulation, and impacts practices. Scientific uncertainties drive the way the optimization of protection has been implemented for many years, very often interpreted as minimization of radiological exposure. And uncertainties make radiation risk communication to the public difficult and sometimes extremely difficult. So to address this issue, the high-level group on low-dose research is action-oriented to accomplish its vision. So the main actions are listed here. First of all, make the best use of the enormous volume of existing knowledge, including that on the variation of risk according to individual characteristics, identify gaps and prioritize them, assemble the knowledge in a consistent manner, promote mechanism-based approach for less uncertain inference from high dose or dose rates to low doses dose rates, integrate biological evidence on radiation-induced cancers, as well as on non-cancer effects with epidemiological results, so that at the end of the day, we are able to leverage the efficiency of research, including the financial strategy. And also, we should improve communication of research results for the benefit of policymakers. So we believe that as a global networking initiative, the high-level group on low-dose research established by the NEA can contribute to and make progress on these actions. With this in mind, the high-level group has created three topical groups that are implementing these three activities. First of all, the creation of an online low-dose research database. Second, there are also examination of the usefulness of the so-called adverse outcome pathway framework jointly with the chemical toxicity community. And finally, development of a policy-oriented and promotional communication strategy. So if we go through the main objectives and activities of the low dose of the group dealing with the low dose research database. So the main aim is to enhance visibility of ongoing and future research projects, promoting networking and collaboration worldwide. So the ideas are to focus on current and in-planning studies in the low dose and low dose rate ranges, offering a broad coverage for a large diversity of research projects. We know that radiological protection needs a lot of different sciences to go ahead. And just asking the principal investigators of those projects a simple description of their project with keywords, the main features, obviously their contact details, and potentially links to access to additional information. So we believe that with this database, we will be able to facilitate proactive networking and collaboration among researchers, and that this tool will serve as a kind of user-friendly catalog to search such projects worldwide. And this, at the end of the day, will help to keep governmental bodies informed on major advances of sciences, and will also help decision-making in funding research. The other activity deals with the exploration of the adverse outcome pathways in the low dose rate area. You may know that the AOP approach was initially developed by the OECD to improve environmental and health risk regulation for chemicals. And the AOP helps structuring published evidence of mechanisms of toxicities spanning multiple levels of biological organization from the molecular interaction to the disease. So it is believed that existing data on biological mechanisms on low dose biological effects can be better organized to improve the understanding of the causal pathways between radiation exposure and health risk at low dose and dose rates, and consequently reduce the current knowledge limitation. Finally, the third activities on the table of the high-level group at that stage is the development of a policy-oriented and promotional communication strategy. The idea there is to clearly define the problem, list the issue of concerns in the low-dose area that would benefit from clearer communication, identify available data and tools that can help make communication more effective, and also exercise how to translate technical results into policy-oriented messages. And it is felt that these activities will contribute to creating a kind of fast track between research results and science-based policies and regulation. So this group will also develop actions to promote engagement of funding organization with by the identification of ad hoc communication strategy for engaging national and regional funding organization, also develop strategy for identifying and prioritizing research gaps. And we believe that the AOP framework and all the related works for radiation-induced AOPs and the low-dose research database will help to do so. And also the high-level group will help by promoting multilateral collaboration to undertake joint research projects worldwide. So to finish my talk, I would like to just say briefly that the high-level low-dose research group will facilitate global networking of low-dose research funding organizations and research implementing organizations, develop a policy-oriented communication strategy on risk uncertainties. With this, we expect that there will be an enhancement of the impact of research, and that at the end of the day will have implication for radiological protection policy, regulation, and implementation. Obviously, also implication for the revision of the ICRP system. Before finishing, I'm pleased to say that the group is currently composed of about 60 experts from 13 countries, invitees from international organization or regional research association. And we are also very honored to have excellent people involved in the governance of the 3 topical groups. They are listed here at the bottom of my slide. And really before closing my talk, I would like to address warm thanks to all the members for their amazing contribution in this very ambitious program of work we have now on our table. So with this, I thank you very much for your attention. I can't hear you, Dominic, you are muted.
Sorry, much better like this, I think.
Yeah.
Thank you very much for your presentation. I was saying that I think we have time to take one question. Very interesting program of work, this high-level group on low-dose research. What are, according to you, the approaches and mechanisms that can be used by this high-level group or any ICRP-PH to interact with the ICRP to provide input in the revision of the radiological protection system?
Okay, thank you for this question. Actually, the mechanisms are quite simple. The first one is that there is a number of members of the high-level group that are also members of the ICRP, so that really helps cross-fertilization in between the 2 aspects. The other point is that the high-level group is operating under the Committee on Radiological Protection and Public Health, and that we have regular meeting with all the different topical expert groups, this one being one among others, and this provide opportunity to present the results wider, so to the all the delegates of the committee. And we have also interaction with another expert group that has been re-established on purpose for the revision of the ICRP system, which is the expert group on international recommendations, where we will review different hot topics. And with this, we have other mechanisms such as creating a new task group dedicated to a specific topics with ad hoc experts. We can also implement topical workshop jointly with the ICRP and others international organization or association. So, and last but not least, we have also, I would say, a rapid and efficient channel to reach our colleagues from the OECD. And this is, for example, of very high added value for the work on the exploration of the usefulness of the adverse outcome pathway framework for the benefit of the radiation research and or its regulatory use.
Thank you, Jacqueline. We have additional questions, but we're going to keep it for the general discussion at the end. And we are now moving to the next presenter. Yuichi Tsunoyama is an assistant professor of the Kyoto University. His main research topic is biological effects of radiation, focusing on dose rate. And in addition to his main research, he has been continuously conducting research activities in the Fukushima prefecture. And the objective of his research group is into improving the modeling of dose rate effect and incorporating it in the field of radiation protection with the expectation to avoid overestimation of low dose and low dose rate exposure effects. So Yuichi, the floor is yours.
Thank you very much, Chairman. Can you hear me? I'm very honored to be able to present our work here. from now, I would like to talk about our sincere hope that newer mathematical models will be taken into account in the next radiation protection concept that you all will build in the next decade. Oops, next slide. As you all know very well, in the field of radiation protection today, the most important and basic mathematical model for assessing stochastic effects is LNT model, LQ model, and these derivatives. The process of deriving the value of DDRVF was based on the LQ model. The linear quadratic model does not include a term for time in its formula. Of course, in the case of one-time extremely high doses exposure, these conventional models works quite well and are really reliable. However, for some exposure situations, it is also a well-known fact that it is impossible to properly assess risk or make precise risk predictions when we use these models. To date, we Japanese have invested trillions of yen for decontamination and treating the highly contaminated water, etc. These huge social and economic losses in Japan are due to the fact that science has not been able to provide proper answers for the long-term low dose rate exposure. And in the field of cancer treatment, this is the case of fractionated high dose exposure. Sometimes the currently used LQ model-based irradiation plan is not sufficient. Concerning this issue, please be sure to watch Dr. Masako Bando's on-demand presentation. And also, at present days, we can travel into space if we want, but we are very afraid that the current approach may lead to a kind of suppression of the entire space industry in near future. So today, I would like to ask everyone here to forget about the LNT model for a moment and think about other possibilities. As an example of this kind of effort, I would like to introduce a new mathematical model that we are currently working on. In recent years, we have proposed a whack-a-mole model, one model, which can reproduce precisely the experimental results on genetic effects in various animals and plants. The core idea of our model is just two things. The first is that a pool of mutant cells is assumed to be the bottom like this, and also like this. And the second is the term of time in our formula, time t. As a result, we can now calculate the genetic effect in also the cases that those changes from time to time. It may be difficult to understand immediately only with this mathematical formula. So please see a short animation movie helping you understand the concept of our model. Okay, here I would like to introduce William Russell's experiment, which was the first time in the world to show that there is a dose rate effect also in mammals. They irradiated gamma rays or X-rays of these dose rates for a long time to male mice and mated with non-irradiated female mice. And then they measured the frequency of mutations expressed in their offspring. You can see a clear difference in the mutant frequency between high and low dose rates exposure. Left graph shows the result of comparison between the values calculated by our proposal 1 model and the actual experimental data of Russell. The 1 model was in good agreement with his mice data. Also, in radiation experiments onto other plants and animals, 1 model reproduces the experimental results quite well. These graphs are 2 examples of prediction using 1 model. The upper graph shows the difference in mutation frequency by dose rate in mice. As the dose rate increases, the mutation rate increases, but it will eventually reach a steady state at any dose rate. The graph below shows a case of fractionated irradiation. The blue line is calculated by use of LQ linear quadratic model, and the red is for the 1 model. Perhaps you can see the length of the cooling time here, and here, and here. And also the length of time after the irradiation completed here are very important if you will perform such experiment. This is a 3D graph that allows you to see the total dose rate, and mutation frequency all at once. There is an equilibrium state of increase and decrease. Currently, the 1 model is now being tested in mice and human culture cells using whole genome sequencing techniques with the support of our government. Through this study, we aim to improve the prediction accuracy of the 1 model. Then, for cancer treatment therapy, we have already constructed the CSO 1 model. This modified version of model included cell proliferation effect and organ volume effect. We are now testing this model on cancer patients at the Osaka International Cancer Institute. Well, in the last part of my presentation, although still a primitive study, I'd like to show that the 1 model has the potential to be applied to evaluate the unstable chromosome aberrations. It is a very well-known fact that when exposed to high doses of radiation, these chromosome aberrations increase in proportion to the exposed dose. Among these, unstable aberrations cannot be duplicated because of their chromosomal structure. For this reason, the number of cells with such aberrations decreases with time-dependent manner after exposure. The effects increase with radiation exposure and then disappear with time, so that it means that 1 model should be applicable to this case as well. It is enough to replace the occurrence and disappearance of mutant cells in the 1 model with the expression and elimination of the cells possessing unstable chromosomal aberrations. Unfortunately, concerning A0 and A1, we have not yet been able to find any measurement data at different dose rates. On the other hand, for B0 and B1, some measuring data already exists. This is the work of Buxton et al. In 1978. They continued to measure changes in the frequency of chromosome aberration in the lymphocytes of patients treated with radiation therapy for several decades. As you can see, stable chromosome aberrations remain for a long time like this, while unstable ones decrease exponentially. Of course, it can be approximated with a simple exponential function, but we try to fit it into a derivative of the one model. The result is this red line. We can fit almost perfectly in this way. Next, we examine this derivative of the one model could also be applied to the astronauts who have completed multiple space missions. Concerning this, variable data has already been reported by Durante et al. In 2003. These are replotted graphs showing the previous slide. The horizontal axis is the time since the first blood sampling, and the vertical axis is the number of dicentric chromosomes per 1,000 of lymphocytes. Gray colored area is the time they are spent in space. When the reduction curves calculated by our model are added on these plots, it seems to be some points agreement between our theoretical values and the actual measurement data, but some points do not agree at all. Unfortunately, we have not been able to find any data where blood samples were taken regularly during long periods in space. So we can only draw an imaginary curve of increasement of dicentrics. From these astronauts' data, authors have drawn very interesting three conclusions. The first is that the frequency of dicentrics significantly increased only on the first flight and then decreased in the interval. Second, the number of unstable dicentrics never accumulates, even after repeated flights. This means also clearly indicate the conventional model using total dose does not work at all. And the third one is the most interesting. Dicentrics showed an apparent decreasing during repeated space flights. Concerning this phenomena, they mentioned the involvement of immunity. If this is the truth, we have to incorporate this newer effect into our model. For example, if we add a correction term to the equation so that the increasing rate of aberrations is suppressed with each space flight, the results obtained by our calculation will be shown like these blue lines. In any way, there is still a lot of room for research as for now. This is the final slide. In exposure situations where conventional models cannot be applied, newer some models that can trace the time evolution of the effects should be utilized. Most importantly, such a model should be constructed with differential equations that can account for those rate effects. Probably, as you know, it is common practice to use differential equations in general physical simulations, such as the calculation and prediction of stock prices, traffic jam, and the amount of water released from dams, etc. Why in our field have we not been proactive in using differential equations so far. We hope that you will consider including differential equations in the next generation of protective models you are building. That's all. Thank you very much for your kind attention.
Thank you, Yuichi, for your presentation. So clearly, your model raises very important questions for radiation protection about the LNT or the linearity of the relationship and the effect of dose rate. I think we are going to take one question from the chat, and we will come back to this question during the general discussion at the end. The question I received was can the WAM model assume interaction between cells?
I beg your pardon?
Can your model assume interaction of cells?
Interaction of cells? What do you mean?
Interaction between cells.
Between cells. Now, our concept is very simple. There is some black box. There's input and output. Our differential equation represent this balance input and output. Can you understand? Sorry, yeah. I'm not that good at English, so.
Okay, we will come back to this question in the general discussion. It's now time to move to the last presentation. So the presentation will be given by Klaus Troth. Klaus studied medicine in Germany, in Munich, and soon he focused on radiation biology and experimental radiation oncology. He has been a professor of radiation biology in London, and after retirement, he continued with the European Master Course in radiation biology. His main interest lifelong has been research on pathogenic mechanisms and of the development of late normal tissue damage. And so no surprise that his presentation will challenge the distinction between the stochastic effects and the tissue response. So Klaus, the floor is yours.
Thank you. I'm an old man. It's nearly 60 years that I was working as an aspiring radiation oncologist in gynecological radiotherapy and studied at the same time for my MD in radiation biology. Radiation protection at this time was based on the determination of the genetic significant radiation dose. That is, the dose to the gonads multiplied with the expected number of children to be begot by the respective person. This exclusive focus on genetic risk is hard to believe today. I still remember how fascinated I was when Wolfgang Jacobi explained to me the new concept of effective dose, which he had developed for ICRP. From this time on, radiation protection was based on the balance of cancer risk, no longer genetics, from a risk-weighted radiation dose and the expected benefit. This concept is still fundamental to all aspects of radiation protection, never really challenged. But this concept was necessarily based on the radiobiological knowledge of the time, more than half a century ago. A thorough re-evaluation of the principles of radiation protection is overdue. Next slide, please. This applies in particular to the distinction between stochastic and deterministic effects. Wolfgang Jacobi at the time explained it to me this way. Some effects are caused by mutational events in the DNA of single cells, such as genetic effects and cancer, and they are to be called stochastic effects, whereas others are the results of killing many interrelated, interconnected cells, such as the acute radiation syndrome, which is caused by the damage to thousands of bone marrow cells. These effects should then be called deterministic effects. Next slide, please. The difference between single cell effects and multi-cell effects would lead necessarily to differences in the dose dependence of incidence at low doses. Single cell effects are characterized by linear no threshold dose responses, while multi-cell effects can only occur if a dose threshold is exceeded. This fundamental distinction between the pathogenic mechanisms is no longer supported by modern cellular and molecular radiation biology. It's radiobiology of 50 years ago. As a consequence, today, the distinction between stochastic effects and tissue responses is no longer based on the mechanisms. It is based on the absence or the presence of a dose threshold. Dose threshold, how do you prove the absence of an effect? The distinction between stochastic effects and tissue responses does not make radiobiological sense, at least not to a radiobiologist of the old generation. Not if based on pathogenic mechanisms, nor on the proof of thresholds. This distinction cannot be upheld anymore. Yet, abandoning it has important consequences. Thresholds entirely depend on the definition of response criteria. Thank you for the new slide. The same radiation effect may appear as stochastic or deterministic. It only depends on the chosen response criterion. A classical example for the dependence of the shape of the dose dependence is on the test criterion is the question of whether cataracts are stochastic effects or tissue reactions. If clinical criteria, such as vision impairment, is taken, it was common use in the past. Cataracts are rightly classified as tissue reactions. If, however, hypersensitive imaging technologies are taken, which detect minute opacifications without any visual impairment, derived threshold doses are very low, and cataracts might well be classified as stochastic effects. Yet there is another problem with the eyes, which have not been studied properly. Results of thorough ophthalmological investigations of Japanese A-bomb survivors 30 years after they were exposed as children showed, and this was published by RERF quite some time ago, the risk of radiation-induced retinopathy may not, go back please, the risk of radiation-induced retinopathy may be higher than that of cataracts, but the detriment would be immensely greater. Another example, developmental disorders have well been documented among A-bomb survivors who were exposed in utero. Nearly 30 children were diagnosed. Please go back. Yeah, thank you. 30 children were diagnosed with severe mental retardation. 16 of them had been exposed with more than 0.5 gray. The incidence of severe mental retardation depends on the stage of pregnancy, is highest between the 8th and the 15th week, and on dose, showing a clear threshold. Yet, next slide, please. When the same population was tested for intelligence, there was a steady decrease of IQ with increasing dose, compatible with the definition of a stochastic effect. Same problem. In one case, stochastic, in the other case, deterministic, depending on the criteria. It has long been known that the heart is one of the most radiosensitive organs in the body, and this has been one of my major research fields for the last 30, 40 years, particularly in clinical studies on breast cancer patients treated with post-operative radiotherapy of the chest wall. An increase in mortality from heart diseases was recorded, which suggested a risk even at mean heart doses of much less than 1 gray. Next slide. RERF studies on cardiovascular mortality showed a perfect linear no threshold dose-effect relationship. It was therefore proposed by quite a few people to transfer the heart into the category of organs which develop a stochastic radiation effect. In their comprehensive analysis of cardiovascular radiation risk, ICRP publication 118, mentioned several times today, arrived at a best dose threshold for exposure of the heart of 0 gray. But is there such a thing as a specific heart disease which is caused by radiation exposure? In the heart, next slide please, 5 different radiation-induced diseases have been described in patients and in experimental animals. Each of these effects can be initiated by radiation exposure, but each one in different anatomical structures in different parts of the heart. Also, pathology and pathogenesis differ between these 5 different heart diseases, as does their dependence of severity and progression rate on radiation dose and even on age at exposure. All epidemiological studies in radiation protection research do not take this into account. Yet pathogenic mechanisms which lead to tissue responses and which define the dose-response relationship, they are not organ-specific, but symptom-specific. Not only tissue responses of the different organs vary within each organ, also cancer is nothing but a summarizing word for a wide range of diseases with very variable biological and clinical features. It does not make sense to pool all types of cancer of a particular organ, in particular if people of different ages are pooled. One telling example of this problem is the fact that in the childhood cancer survivor studies, 50% of all radiation exposure-related second cancers were sarcomas. That is, the critical tissue is the connective tissue, which does not even feature in the list of tissue weighting factors. Next slide. the distinction between stochastic and deterministic radiation effects has to be abandoned. Also, the concept of radiation detriment does not heal the problem. The concept of radiation detriment involves complex value judgments. The integration of heritable effects presents particularly sensitive issues. Little research has been performed on heritable radiation effects since the pioneering studies of Sankar. Their purely numerical inclusion in the concepts of detriment and effective dose requires comprehensive discussion and new research as well. And I'm very happy to have seen some attempts for that today. The current system is based on the concept of the mutation doubling dose, which in mega experiments on mice had been determined to be 1 gray at low dose rate. However, the 7 locus test, which has been used for these experiments, does not investigate those types of mutations which ICRP used to estimate the risk in children of radiation-exposed parents, namely specific single gene mutations with dominant inheritance. The 7 locus test studies mainly deletions of whole genes or long stretches of DNA. These types of mutations are known in human genetics to be involved in the causation of genomic disorders. Many years ago, Sankar suggested to study specifically the risk of those disorders in the children of exposed parents, and thus create a better base for genetic risk estimation rather than the doubling dose method. And maybe, although I don't expect it, that I see final good results from the Australian studies which have been presented today. I conclude, next slide, please. Next slide, please. The distinction between stochastic effects and tissue reactions is not compatible with current radiobiological evidence. Any attempt to construct a comprehensive system of radiation protection, which includes all radiation-induced health effects in all human beings, is bound not to make sense, at least not to a medic. A new system of radiation protection should not be based on a system of radiation effects in specific organs or tissues, but on specific exposure situations. Thank you.
Slide.
Thank you very much, Klaus, for this very...
thank you for doing the slides for me. or whoever did it.
The thank is to Mike. Thank you very much for this very interesting presentation, which raised a lot of different questions for the current system of radiological protection. Not only the classification of the effects, but also much more than that, about WT and so on. Before going to the there are many aspects that will be discussed in the general discussion, I guess. Before going to that, I would like to come back to one of your slides. The distinction between stochastic effects and tissue reactions must be abandoned, which is very strong, and most of the system is based on that. What do you propose instead of that? Do you think it has to be completely abandoned or adapted to the current knowledge about radiation effects? Could you go a little bit further on that specific point?
My experience from clinical and experimental work is that in specific organs, different types of damage, which could be either classified as stochastic or as deterministic occur. Practically all organs show a wide range of potential radiation effects. So the specific the dose to an organ is important. Even the inhomogeneities play a huge role. And you have seen, depending on where in the heart the dose is maximum, different effects will occur. And they may occur early or they may occur late, 20, 30 years later. So I think we should go back to the basic radiobiological observations and see, is there a fundamental difference between cancer induction, which certainly is not only based on the stochastic effect of the primary lesion. It's a multi-stage process. There's a lot of cell-to-cell interactions, which already would be something like a tissue or effect, or at least a multi-cell effect. So I think we have to consider radiobiology in its full aspects to see, is there such a difference? And the difference between stochastic and deterministic effects is very big, as we've seen in the cataract discussion, where suddenly threshold doses went down nearly by an order of magnitude. So just because of that, on the other hand, as I showed you, we don't really consider all the data, particularly in the eye. These retinopathies in Hiroshima and Nagasaki have only been published once and not even mentioned in the abstract of the paper. This, however, even if maybe it's overstressed, but nevertheless, a retinopathy cannot be cured, leads to blindness. A cataract is a matter of an operation of 10, 15 minutes and a cost of less than $1,000. I think we really should look more at the raw data and not on the models. That's my experience from 60 years radiobiology. But I don't know yet, but I would love to be involved in these discussions. What can be a good replacement for these two? I think we only have to have one endpoint, and that is disease and detriment, and not fundamental differences between one which has a linear no threshold and the other one which has a threshold. This doesn't make sense to me. We don't need it, the threshold.
Thank you very much again. That's clearly an important question for the ICRP and for the evolution of the system in the next years, and clearly a question for Committee 1. As you may know, there is already a task group on cardiovascular disease with this question included in the work of this task group. What is the good classification for this kind of disease? But of course, the question is much larger than that and applies to all the different effects. I think we should now move to the general discussions. So I invite all the speakers to show up. We are perfectly in time, so thanks a lot for each of the 5 of you for keeping the time. And we have 30 minutes of discussion. There have been plenty questions, so we are not going to cover all the different questions, but we will try to make it as diverse as possible. And before beginning this general discussion, I would like to, as it has been said by Nobuhiko and Barry Werner in the previous session, to remember that there are also on-demand presentations that are available on the platform. So don't hesitate to go and have a look at that. There are many of these presentations that are directly related to the effects and to the risk about the AOP, about the LNT, about the uncertainties in cancer risk at low dose, about stem cell competition, about the usefulness of DALI, and also presentation of the ongoing work of different task groups, C1 task group especially. So don't hesitate to go and have a look at this on-demand presentations. And now I would like to begin the discussion. So again, to all the participants, don't hesitate to ask questions on the chat, and we are going to consider it. I would like to begin with a question to John, to the first speaker. There were several questions coming back on doses. So I tried to combine these different questions. One was about what level of doses are you speaking about? To be a real effect, we need a link with between the effect and the increasing dose. And one by Jack Valentin, I imagine most medical exposure will give very low doses to the gonads. What is the real potential for study of any study to find anything at this level of doses? So John, these questions are for you.
Yes, very important questions. I guess we know that most diagnostic doses are low, and it sounds implausible that we would detect an effect. But a detailed look at the literature actually suggests that copy number variants are more sensitive to ionizing radiation than might be suggested from the mouse experiments, and certainly more sensitive than the negative point mutation experiments in the post-Chernobyl study. So I just don't think we can rule out the possibility of real effects. Klaus just showed us the results from intellectual impairment in children exposed in utero to the atomic explosion. And yes, it does look like there's a threshold, but the important information for me is that when you're just looking at some of the outcomes that are now known to be genetically determined via copy number variations, these are the kinds of outcomes that are very significant for families: intellectual disability, congenital abnormalities. And we need to really strengthen the evidence to be able to say they're not due to low dose radiation. I mean, I just, until we've got the data, we can't exclude that possibility. And that's why I think we need to proceed along the lines I outlined in the presentation. I'd be very interested to know if there are others who think the same. I'm not suggesting we know the answer. I'm just saying I'm not logically convinced by all the available evidence that even low doses like 10 milligray can't have an effect.
Thank you, John. Other among the authors, do you want to extend also on the last words, evidence of an effect at low doses? Okay, so maybe we can move to another question from the panel. Maybe we stay with you, John. There were several questions on the population. One was, will you consider only diagnostic exposures, or will radiation therapy will be considered also as a comparison? What is the availability of data for other populations? Can you elaborate on the control group? There could be a bias in the patients compared to the general public. So global questions about the population. Do you want to answer on these different points?
Yes, look, the short paper that I submitted in support of the presentation deals with some of the epidemiological issues we need to address. Very important. And the experience we had with looking at low dose effects from CT scans just says with a large data set, you can pick up effects that will be missed with small data sets, and but it does raise the important point that the nature of the population is very important. We've established the precedent that we can do record linkage to answer questions like this. And there's a wealth of epidemiological expertise that can address the problems. There was a comment about what we do about therapeutic radiation. Well, we're able to identify patients from the health records, and we won't have necessarily good doses for people receiving therapeutic radiation, but we'll be able to look at them separately. And as most of us would know, there won't be large numbers of people of reproductive age who are having therapeutic radiation. So we can obviously look at the question, and really the data will provide the answers, and there are some very good people who are able to deal with some of these data problems already. Yeah.
So before moving to another topic, there is the last one recent question is about we know that pediatric CT studies on brain tumor can have potential biases such as reverse causation, confounding by indication. What will be the potential basis in the planned record linkage study on the next generation effects, and how can they be evaluated or avoided? So any comments on these biases for cancer, John?
Yes, there have been real challenges. And all I can say is that using the best analytic methods, one can investigate what biases are doing to the data. For example, we've used propensity score analysis to try and adjust for confounding. We've looked at finite mixture modeling to look at reverse causation effects, and given good data and given good people to work with the data, these problems can be addressed. And that's basically where I'm at, and not saying that there's necessarily a high probability that these effects have been missed hitherto, but just trying to make the case that the science has got to be driven by data, and we've got to deal with the biases that will inevitably be there. Yeah.
Thank you. Some questions were for you also, Nobuhiko. Especially there were several questions about you proposed to develop an open source software in your presentation. Could you elaborate on that? And the second question on the same topic was, do you think it is the role of the ICRP or of the IEA to develop an open source software to improve consistency and simplicity across different states? So could you elaborate a little bit more on this software?
Okay. Thank you for these questions. And in my experience in the TG102, it was quite hard to reproduce the calculation in publication 103. It was much harder than initially thought. And finally, we had to check the software or script of used in the calculation step by step, and to try to understand what was done. So I do not think this kind of work should be repeated every time. So we should become more, how to say, better approach. So I am proposing to develop an open source software. And from my personal point of view, the team that does calculation for the new recommendations should make their tool open to the public. So that is an ideal way.
Okay, and for the second point, is it the role of the ICRP or the role of another organism? Or?
So, as I said, from my personal point of view, ICRP should make the tool available with the cooperation of the special team that does the calculation.
And another question, which is for you also from Martin Tandel. The National Cancer Institute has identified 19 cancer sites related to exposure to ionizing radiation. Will these or other cancer sites be included in assessing the cancer detriment?
Pardon? Could you repeat?
Yes, the National Cancer Institute has identified 19 cancer sites related to exposure to ionizing radiation. which is larger than what is currently considered in publication 103. Will these or other cancer sites be included in assessing the cancer detriment?
Yeah, actually, as I told, currently in publication 103 model, only 10 cancer site-specific models. So we should expand the coverage as many as possible.
I completely agree. And for information, so there is a subgroup inside Committee 1 dealing with this specific question. What has been published in the literature since 2007 that could be used to improve the modeling of cancer risk? I don't take all the questions. There was a very specific question for you, Jacqueline. I don't know if it was about the low dose research database. In terms of radiobiology, would the low dose research database be needed to capture data from fluoroscopic guided interventions? So can you elaborate about that?
Yeah, I just would say that like the name is indicated of this database is dedicated to only research projects dealing with low dose and low dose rates. So if you are not within this range of low dose or low dose rates, you are not welcome in this database. So the idea is really to make leverage of international cooperation on this difficult issue and to advance more rapidly research, the many research topics on the table.
And another question was about the objectives of the high-level group and low-dose research. What are the most challenging activities for the high-level group and low-dose research to accomplish its vision?
Actually, the three activities I presented of the high-level group are unique and to our knowledge have never been developed before at the international level. So that means that each of them correspond to a challenging gap that the HLG will try to fill, and as such will contribute to the science underpinning the international radiological protection system, as well as the RP-related policy, regulation, and practices. So maybe the most important challenge we should demonstrate quite rapidly is to demonstrate that this group is attractive and can demonstrate easily its added value in terms of the new things on the table. And maybe this is the most challenging point. So to disseminate enough, to be attractive, and to be able to bring quite rapidly kind of success stories on the activities of this group. So I hope we will be successful, at least with the different activities. As I said, it's easy to, if our database increase in terms of occurrence and regularly, and if it's going to be a kind of reference to search for cooperation between researchers, to search whether there is overlapping in planned research by a funding organization. That will be one of such success we are looking for.
Thank you very much. Among the numerous questions, there was one specific one for you, Yuichi, from Agnès François. Do you really consider that the LQM model is not suitable for cancer treatment? It is suitable as well for tissue effects, tumoral and normal tissue, isn't it? So that was a question for you about the advantage of your model and the fact that a linear quadratic model was not suitable for cancer treatment, I guess.
Okay. at present, the biggest challenges for our model is the risk of cancer development. We succeed to reproduce the genetic effect, and also we have already succeeded in building a CSO1 model that agrees very well with the results of treatment, at least for the process of shrinking a tumor that has grown to a certain size by radiation. However, how should we define cancer as a disease? Is it the size of the tumor? Should the grade of the tumor be taken into account? How should metastasis be considered, etc.? There are still a lot of issues to be solved in order to make a mathematical formula for the cancer risk. This is very big issue, but we are trying to improve now.
And I had a question for you also. I remember your approach tries to model the effect of dose rate effect and not the effect of cumulative dose or dose, which is similar in my view to the approach that was used in mechanistic models. You know, like two-step clonal expansion models, for example. What is the advantage of your approach compared to these types of mechanistic models, which were also modeling the impact of dose rate effect?
I don't know quite well, but it's my opinion. Our model is as for the data on the genetic effects of radiated experiments, it's plants and animals. Our model gives very good results. What we would like to emphasize most is that the time component should be properly taken into account in the formula, and maybe this is the most different former some equations and models, I think.
Okay, and one additional question, which is for you, but also for the panelists. I think will introducing of more complex models like the WAM model, more radiobiologically right approaches like non-categorized effects improve the applicability of practical radiation protection. To summarize, is it because we are going to use more elaborated models that we are going to have a system which is more applicable to radiation protection?
I can only the proposal of mathematical boring model like now, and then I hope we keep continue to arguing how to improve this model and how to adjust into the protection, newer protection concept, I guess.
And also on this topic of the effect of dose rate and low dose rate, there is a task group, Committee 1 task group on dose and dose rate effectiveness factor. And there is a presentation in the on-demand presentations that gives some insight on that. So don't hesitate if you want to have more information about dose rate. Don't hesitate to go and look at this presentation. And to finish, I would come back to you, Klaus. So there is a question which is for you, maybe not only for you, of course. should the system of radiological protection assume that there will be never any medical progress in treating or preventing harmful effects of radiation? That assumption is implicit in risk assessment for long-term management of radioactive waste, but not only. So could you develop on this question? Also the other panelists, I guess.
I think we know already a lot, but somehow I have the feeling that the radiation protection community, of which I am a part, is not considering all data. The situation is more complex than modeling ever will be able to show. Moreover, there are sources of information which are completely neglected, and I showed you the data on the effects on the eye. Moreover, radiotherapy is often neglected by radiation protection, but you should not forget radiotherapy means high doses to a few hundred grams and low doses to 70 kilograms. So from second cancers and other so-called abscopal effects of radiation in radiotherapy, we could get a lot of information. I think we should go back to the raw data, and I think we can predict effects of low doses, in particular with regard to these very long decay times of radioactive waste. What's the difference to the heterogeneity and variability of natural background radiation? The sources are similar and the doses are similar. Why not the effects? I think that good studies on the heterogeneity of natural background radiation based on various modifying factors are a much more convincing source of communication with the public than risk estimates. Risk estimates are good for specific exposed populations like workers or patients in diagnostic situations, in radiology, maybe even in radiotherapy, but I don't think there are good sources of information for the general public, in particular not for the radioactive waste situation. It's different for accidents, and Chernobyl has shown that. But even there, we learned a lot, but a lot of what has been said is certainly not scientifically valid. Thank you.
Thank you very much. We are at the end of this session. So again, I would like to thank all the participants for the questions, and there are many questions that have not been discussed and answered. So I will check with Chris how we can manage listing these questions and providing answers. And thanks again to the 5 panelists for their very interesting presentation and the very interesting discussion about these different things. Clearly, the discussion has been dealing with questions that are very important for the current system about the classification of the effects, about the heritable effects, about the shape of the dose-risk relationship, about the effect of dose rate, about the understanding and the presentation of the mechanisms and the AOP. So all points have not been. There was no time enough to go in details in each of the points, but that's really questions that are very interesting for the evolution of the radiation protection system. So thank you again for the quality of the discussion. with this, I think it's time to end up with this session number 2. So again, thanks to all of the participants. We reached more than 400 participants in this session, I think. And so I would like to welcome you for the session 3 and session 4 tomorrow, and for the continuation of this open discussion regarding the evolution of the radiation protection system in the next years. So thanks a lot, and with this I would like to close the session.
And thanks to Mike. Yeah.
Thanks, and thank you again for the 5 speakers.
Thanks
And thanks to Mike for the organization. Yeah.